couple of recent methods that we've developed to identify regulation or diversity in regulation based on high-throughput data. Okay, so like most people in this room, I'm interested in being able to annotate the functional genome, especially the non-coding part. And as a community, I think we are getting closer to doing that, thanks to the advances in high-throughput technologies, which when coupled with really clever assays, can identify different regulatory and biochemical activities along the genome, right? And one such activity is protein DNA binding. And an assay that does that and is popularly used is the chromatin immunoprecipitation assay, right? So you begin with live cells, treat them with formaldehyde so that cross-links protein-protein and protein-DNA interactions. You then lyse open the cells, cut up the DNA, and immunoprecipitate all those regions associated with a protein of your interest with an antibody against it, right? And then you do a control experiment also. So after reversing the crosslinks, you get 2 sets of DNA, which you then sequence and align back to the genome to get peaks, which tell you where the protein is binding. Okay, now transcription factors typically bind tiny regions between 5 and 20 base pairs long. The peaks, however, are larger, right? At least 100 base pairs usually, and sometimes all the way up to 2,000. Okay, and so what is typically done after you get these nice plots is you take all the DNA sequences at the peaks and try to find an enriched motif in the set, right? So the assumption is that since all these sequences were reported in the same experiment for the same protein, they must contain a binding site. Maybe not each sequence, but at least the binding site or the motif must be enriched, right? And you don't stop at one. You find all the occurrences, mask them, do it again, and you keep sort of reporting motifs in enrichment order. And there are many methods that do this. And they do this in different ways. Some use additional information like conservation or an unbound set or some information about where the protein might be binding in the center or something. But they essentially use the same criteria that the motifs that they return must be enriched in the full set and kind of independently of each other, right? And this sometimes causes difficulties when the biologist knows what motif to expect, right? Because usually they work with transcription factors they know the binding sites of or specificities of, and they find that the motif is coming maybe at number 3, number 10, not at all, or is accounting for a very small fraction of sequences that were pulled down. And it becomes a little less satisfying, right? So, so I think this stems from the fact that we are still treating the chip experiment like a black box. So the sequences that come from it are just treated like all sequences in one shot, like the same. Okay? But we do know a little bit about how a chip experiment is done, right? So let's say in this cartoon, we are profiling the red pentagon, right? So that's the protein that you have an antibody for. So after the experiment, you're going to pull down all complexes involving the protein, right? And of course, some DNA, because you are sequencing eventually. And in this cartoon, you should be identifying 4 different complexes, pulling down 4 different complexes. But what you're actually getting from the set is the DNA sequences, which are a miscellaneous set, right? It's a mixture of all these complexes, right? And what we have is a method called diversity that tries to identify the diverse components of this mixture. Essentially, it says that there must be one cause or one reason for that sequence to have been reported in the experiment, and that cause is modeled as a motif. Okay? So in traditional motif discovery, you have your set of sequences, that's the same, and you're trying to find a motif that's enriched in the full set, as well as the positions which contributed to the motif. In diversity, you are actually splitting the set, okay, into k different causes while simultaneously identifying one motif in each set, right? And so you're learning a few more things now. You're learning simultaneously k different motif parameters. The positions, of course, those are the same, like in this case. You're also learning an indicator that kind of tells you which set each sequence belongs to, right? Which motif it contributes to. Okay, so I'm not going to talk more about the method, but it's a probabilistic model, and we use Gibbs sampling to learn it. And the method is sort of described in bioRxiv and also in an earlier paper in nucleic acids. But I'm going to show you some recent results with that. Okay, so we looked at an insulator binding protein in the fly. Okay, it's one of many insulator binding proteins, CTCF. And this is from Kevin White's lab, where they profiled it in 4 different fly species in an early developmental stage. And they found that, yeah, the CTCF motif was enriched, right, in the traditional sense, in the bound regions. And they also said that there was high divergence in where the protein was binding, right, based on the chip peaks across the 4 species. Okay, so this is what the input to diversity looks like. Okay, so this matrix is an image where every row is a DNA sequence and color-coded according to the nucleotides. Right? And what we're doing, if you remember, is partitioning the set. Okay, so after running diversity, it says that there are 8 optimal causes. Okay, so 8 partitions. Okay, and this is the center of each partition, is noted here as a motif. Okay, so you'll notice that the first 4 motifs have a very strong resemblance to the fly CTCF, but there are variations. Okay, and one of them, in fact, looks a lot like the human binding specificity. And what we also noticed, what you can do next is look at all the different characteristics for every sequence in each motif and start figuring out what these motifs might mean. Okay, so first we looked at the chip score, essentially the peak height. And so the first 2 have a significantly higher peak score, the first 2 variants of CTCF. The human one, for some reason, has a weaker, lower score. And then these guys are probably cofactors. They're not direct binding sites. And so they have a lower score. But what you can see is the diversity that you see in sequence kind of correlates with the diversity in the chip score. Okay? And so we looked, so these 3 motifs are well-known motifs of a few transcription factors. This is the promoter element, OLR1, I think, or one of them. And we looked at overlaps with other ChIP-seq data, and that sort of fits well. Okay. We also looked at where the motifs were along the genome, and we noticed that the CTCF ones, the direct binding ones, were more variable in where they were placed as compared to, of course, the promoter element, thankfully, is really, really close to transcription start sites. Okay, and we finally also noted that there was diversity in evolution as well at these modes. So the CTCF modes, the direct binding sites are highly conserved. The pink just means it's more conserved. And PETA, on the other hand, which is probably a cofactor, is not. Okay, and so we also ran diversity on the other fly sets, and we did not find PETA come up. Okay, so there's probably more divergence in the cofactors and not so much in the direct binding regions. Okay, so if you have your own data set, you can try out our web server, or you can also download a standalone version that's on GitHub. Okay, so I'm going to move on to a more recent method that we have. Okay, so this works okay for ChIP-seq data, where you can imagine that there is one binding region that is a cause for reporting. Okay, but what happens when you have other data, like DNase hypersensitivity sites, where you're profiling open regions, where the cause of being reported could be a set of motifs, right? Particularly because you are identifying promoters, enhancers, and all sorts of things that are open, right? Similarly, for high-throughput enhancer screens that we just heard about, right? You could have multiple motifs defining the diversity that you see, okay? So what I'm proposing is the following. So we now start off again with motifs, okay? So the model says that, yeah, there are motifs in the set, but they act together in modules. Okay, nothing new in that. And also, you don't expect only one instance of a module to appear, right? So there must be multiple occurrences. But of course, when you are profiling the data, or when whatever experiment you're doing, you're getting, again, the mixture of all these sets, right? So you're just getting the DNA, and we are trying to work out what these modules would be. Again, we are not looking at a module that's enriched in the full set. We are looking for a set of possibly diverse modules that explain the set, right? And again, they are based on these motifs, which also we do not know anything about, okay? So this we call module diversity. So what we are saying now, as compared to diversity, where we had one motif per partition, we have now a set of motifs per partition, which are the three colors that you see here. And each partition is essentially some sort of a combination of the motifs, okay? The phi's are the motifs, okay? And the hope is that most of the motifs are irrelevant, okay? And I've put it as a addition, but it's not really. It's a probabilistic model again, okay? So we are learning motifs. We are learning the modules, the indicators. We are also learning, so therefore we are learning the different parameters of the motifs. We are learning now the position of every motif in every sequence. Many of them would not be there in a sequence, okay? And then you have the module identity, which is the colors. Okay, we have a probabilistic model for this as well. So instead of addition, it's actually a product of Bernoulli trials or Bernoulli distributions. So you either have a yes or a no, whether a motif is present or not. Okay, so I'm going to show you some results with this. This is ongoing work, right? So this is again ChIP-seq data for estrogen receptor. Okay, now this protein is active in cancer cell lines, and in this paper they showed that it had different cofactors probably in two different cell lines. Okay, and this is how they got the motifs. The motifs were identified after the EREs, which are the known direct binding sites, were masked out. Okay, the traditional way of finding motifs. Okay, so when we applied module diversity to this, okay, which were around 5,000 sequences, okay, we get this. I'm going to explain what this means. Okay, so the bands, the 10 bands that you see are the 10 motifs that it learns. Okay, those are shown here. And it learns some 9 different modules, okay? And a black color just means that that motif was not present, did not contribute to what you see here, okay? So all these lines contribute to motif 8, okay? If there's a black one, it's just not present, right? And so what was reassuring, of course, that you do get the ERE, right? This is the full ERE site, this is the half site. Oh, sorry, this is the full one, and number 4 is the half site. Okay, and we also find the ETS family motif, okay, which is out here. Okay, but it comes in 2 different modules. So if you look at the 2 modules separately, right, you see that it's either occurring with a half site, or it's occurring with all these sprinkles of other motifs, which everything, let me remind you, is done de novo, right? No information about the motifs is given at all. Okay. So that just probably indicates that even the tethering model probably has 2 different ways of being active. Okay, so that was ChIP-seq. Not very interesting because we just saw diversity also on ChIP-seq. This is on DNA's hypersensitivity sites. Okay, so this is a big set. Okay, it still finds lots of modules. And let me just show you in the interest of time. 3 modules that seem to be closest to the transcription start site, which this one is really the closest. And when you look at the motifs, you'll actually see identical motifs, almost identical motifs come up. And I was a little confused to that because we don't have any number. You remember, it was a Bernoulli trial. You either have the motif or not. So what this is actually saying is that you need 2 occurrences. So it's automatically coming up. For this module, you need 2 of them. And then you also have other motifs that show up, which are known promoter elements, at least some of them. And this is what the scenario looks like on the 3 modules that are really far away. Okay, so you see variants of CTCF and really nothing else coming along. Okay, all right, so this I could not resist because we had a keynote speaker today talking about his work. So this is star-seq data, which is an enhancer screening data set, and they did this in 3 different cell lines, and they showed that there were some motifs that were enriched in one cell type, some motifs that were enriched in all. Okay, this is by looking at intersections of the enhancers that you get, and then comparing them with the shared and non-shared and things like that. Okay, so this is what you get when you just run it on the S2 data set. Okay, you get a lot of motifs, okay, but these are enhancers, so maybe that is expected. Okay, and then you can start asking questions about whether these modules do intersect with the other enhancers. When you do that, you see that there is a big module that is S2 specific, and there's another one that is common. Okay, so of course they're not all in there, but just if you do a hypergeometric p-value, that's significantly in other cell type specific and significantly common. And as you can see that there are some that are just some bars or some motifs that are common, but there are also some that are specific to the cell type specific ones, which are these guys. Again, no knowledge of the GATA or anything was given to it. It's automatically finding these motifs which are, I think, known to be or expected to be active there. On the other hand, you get the repetitive regions automatically coming up in the modules that are shared across the cell types. Okay, so hope I've convinced you that there is a lot of diversity in the same experiment, data from the same experiment that you might, from a single experiment, and you can actually decipher it in terms of motifs and modules without any prior information, without a priori selecting for any property. I do think that the property should come out automatically from the sequence, because after all, it's when the motifs when the transcription factors are acting together, they're not looking at the property beforehand when they bind there, right? It's the sequence that is giving it the activity, right? So with that, I'll end by thanking Sneha and Anusha, who were instrumental in diversity. Rahul is my collaborator, and we also have a poster, so I encourage you to go and look at it. It's on a different problem, and so we are looking at detecting motifs very quickly in large data sets by using a very novel clustering approach. And thank you for your attention.
We have time for one quick question, if there is one. Okay. Thank you.
Thank you.